This is part 2 of the chapter 16 recordings, and we are going to talk about DNA replication. So we've learned that DNA replicates during the S phase of interphase. So DNA always makes a copy of itself so that the single-stranded chromosomes become double-stranded with 2 sister chromatids. And that happens right before cell division, right before a cell divides. The original strand of DNA, we call that the parent strand. And this is basically how it works. If this is our parent strand, number one, I want to point out to you that if we only could see the left side, if we could only see the A, C, T, A, G, we would automatically know what the right strand code was because, so let's say that on the left strand, we call that the template strand, and then the right strand, we'll call it the complementary strand, because that's really the way you should say it. So we would always know the complementary strand if we just know the template strand, because we know that A always pairs with T and C always pairs with G. So if this is A, we know that on the right side would be T. And if C is on the left, G would be on the right. And so that's how, we always know the sequence of the complementary strand if we know the sequence of the original template strand. But this is the parental DNA molecule, right? It's double-stranded, and it's going to make a copy. It's going to replicate. So what it does is it unwinds and unzips, so the 2 strands separate. Separation of the parental strands. Now each strand is a template strand, and a complementary strand is added to each of the old parental strands. So nucleotides are added to their complementary base. They're paired with the one that, based on Chargaff's rules. And so you end up with 2 new strands. These are going to be what forms the 2 sister chromatids in our double-stranded chromosome. So we've got 1 old and 1 new strand in each of our new DNA, replicated DNA molecules. And so we say that this process is semi-conservative. It conserves half the old. We call it the Watson and Crick discovered the semi-conservative model of DNA replication. And here you can see it again. The semi-conservative model means that you're going to end up with, you would have a parental strand, which is original DNA. Then it would replicate. You'd have 2 strands that had half old and half new DNA. And the next replication, you would end up with, if you were to analyze the chemistry of what, all 4 of these, you would end up with basically 1/4. Yeah, you would, if you look at the amount of DNA that is blue from the original DNA strand, you'd have 1/4. After the second replication, you'd have 1/4 or 25% of the DNA would be from the original parent strand. And then it would just be less and less over time. But after the first replication, you'd have 50% of the DNA from the parent strand. And after the second replication, 25% would have come from the parent strand. So the semi-conservative model wins. And there was an experiment conducted by Meselson and Stahl where they did analyze the composition of the DNA after 1 replication and then after 2 replications, and they did find that the DNA sample after 1 replication was 50% from the original DNA. After 2 replications, it was 25% from the original DNA, and that supported the semi-conservative model. So we are going to look at DNA replication, and we're not going to study DNA replication in bacteria, although there's some information here about it. But we are going to learn about DNA replication just in general, like how the parental DNA replicates and forms two identical sister chromatids. Okay, so the first thing that has to happen is the double helix has to be separated. The two DNA strands have to be separated. So there are enzymes that do this. They are called helicases. Helicases, enzymes that separate the helix. Enzymes are always named for what they do. So helicase is the enzyme that untwists the double helix and separates it. And then so that it won't reattach, because it's kind of being pulled back together, like partly those bases that were the complementary to one another are kind of wanting to bind with each other, just by their nature. So single strand binding proteins will bind and keep the two parent DNA strands separate from each other, okay? And then we have topoisomerase, which relieves the strain of twisting by breaking, swiveling, and rejoining DNA strands. And I don't really understand that, but I do know helicase separates the strands. That's this green enzyme. Single-stranded binding proteins keep it from sealing, back together. And then topoisomerase is there for stability. Primase is an enzyme that binds and starts the replication of the one of the old strands. So the enzyme that comes in, you don't see it in this picture, but there's an enzyme that comes in after helicase separates the two strands. There's an enzyme that comes in and adds nucleotides to each of the old template strands, and it's called DNA polymerase. But DNA polymerase can't do its job. It can't start building the new strands until a primer, until a primer and primase, they play a role first. So the enzyme primase synthesizes a short nucleotide chain, and it's actually an RNA, not DNA. So primase synthesizes a short RNA primer, and that primer will bind to the parent DNA strand, and then that will enable DNA polymerase to begin adding nucleotides. Okay, so most DNA polymerases require a primer, which is that RNA strand that begins DNA replication, and they require a DNA template strand. So going back to the picture, the primase enzyme synthesizes the RNA primer, which is a few nucleotides long, and once that is synthesized, DNA polymerase can come in and start adding nucleotides to the old strands. And that's pretty much how the basics of how DNA replication works. All right, you can see a little more of that here. You can see that DNA polymerase is coming in and it is adding nucleotides to the old strand, one at a time. It's actually a pretty quick, fast process. Now, there are two old strands of DNA, and we call them template strands. But one of them runs in one direction and the other runs in the other direction. And the way we describe directions in DNA is we use the numbers 3' and 5'. 3' and 5'. And DNA replication runs from the 3' to the 5' direction. Okay, DNA replicates in the 3' to 5' Okay, and it's not worded that way. Let's come back. I'm sorry. All right. The nucleotides can only be added to the 3' end of the old strand, and then they would be added to the 3' end, and they would be added moving toward the 5' end. But the way we word that is DNA is replicated in the 5' to 3' direction. That is the replication, the direction of replication. From the 5' end to the 3' end. DNA is replicated from 5' to 3' in that direction. So there's a problem with that. One of our template DNA strands, called the leading strand, runs in the 3' to 5' direction, so it can be replicated. We say it's replicated continuously from one end to the other. But the other strand is called the lagging strand, and it runs in the opposite direction. The lagging strand runs in the 5' to 3' direction. So because it runs anti-parallel to the leading strand, it can't be replicated continuously. It's only replicated in pieces. So basically how that works is the lagging strand has to be replicated in smaller pieces, not continuously, not in one long piece. The DNA polymerase must work in the wrong direction with the lagging strand. So the lagging strand is synthesized as a series of segments called Okazaki fragments. And those fragments are joined by an enzyme called DNA ligase. So now if we go back, we can look. All right. If you look at the original parental DNA, we know that helicase is the enzyme that separated those two parental DNA strands. We now call them template strands. Single-stranded binding proteins will keep those strands from resealing or coming back together. DNA polymerase is going to add nucleotides to the leading strand in the 5' to 3' direction, and then it's going to add into the lagging strand in Okazaki fragments, and then the fragments will be sealed together by DNA ligase. And I would watch these two animations. The animation on the synthesis of the leading strand is slide 52. Synthesis of the lagging strand is slide 55. And then an overview of DNA replication is slides 56 and 57. And here we have a summary that puts it all together. Again, the beginning is DNA helicase separates the two parental DNA template strands. All right. And then single-stranded binding proteins hold open those two strands so they won't reseal. Now, the leading strand, the leading template strand, the synthesis of the new strand that's complementary to that template strand, that starts with an RNA primer. And then DNA polymerase comes in and adds nucleotides continuously from one end to the other end in the 5' to 3' direction. And so now you end up with one of your new DNA strands, which is half old and half new. And then the lagging strand, DNA polymerase comes in and synthesizes segments of that, one at a time. And we call those segments Okazaki fragments. Then the Okazaki fragments are sealed back together with DNA ligase. And now you have your second new DNA strand, which is half old, half new. And DNA replication is complete. Here are all the enzymes involved. And the only thing is I didn't really go into the difference between DNA polymerase 1, 2, and 3. Okay, there are DNA polymerase 1, 2, and 3. So this is a good table because if you learn the enzymes, that kind of helps you learn the steps. This is bacterial DNA replication, proteins and their functions, and the replication is very similar in really any species. You're going to have helicase, which unwinds the parental strands and separates them. You're going to have single-stranded binding proteins that keep those parental strands apart. Topoisomerase, which relieves overwinding strain ahead of replication forks by breaking, swiveling, and rejoining DNA strands. So the topoisomerase rejoins the DNA strands once after they've been separated, once they're no longer being copied anymore. All right, then we have primase, which is the enzyme that synthesizes the RNA primer. And the primer is synthesized at the beginning or at the 5' end of the leading strand, and then it's synthesized at the 5' end of each of the Okazaki fragments of the lagging strand. So the RNA primer has to always be the beginning of every new or complementary strand that is added to the original parent strand. So you have to have RNA primer at the beginning of the leading strand. Then you've got to have RNA primer at the beginning of every Okazaki fragment. Okay? Then there's DNA polymerase 3, which adds nucleotides to an RNA primer or pre-existing DNA strand. Then there's DNA polymerase 1, which removes the RNA nucleotides of the primer and replaces them with DNA nucleotides. And DNA ligase that joins Okazaki fragments. Okay, now sometimes we have mistakes that are made during DNA replication. And so another role of DNA polymerase is to proofread the new DNA, the newly made DNA. And when it detects mistakes, when it detects incorrect nucleotides, it will replace them. Then you have, even after that, you have a process called mismatch repair, where repair enzymes replace the incorrectly paired nucleotides that got past the proofreading process. So DNA polymerase can proofread, and then we have other repair enzymes that will repair anything that didn't get repaired by the proofreading. That's called mismatch repair. All right, then we can have sometimes chemicals and exposure to X-ray radiation or other radiation, exposure to harmful substances or conditions in the environment that can damage DNA, and it can cause mutations in the DNA, which are just changes in the DNA code. So there is another type of repair called nucleotide excision repair, and that uses an enzyme called nuclease. Nuclease cuts out the damaged stretches of DNA and then replaces them. So this is called nucleotide excision repair. So we've got a mistake here in the DNA. We've got a nuclease that comes in and removes the mistake, and then DNA polymerase comes in and replaces the incorrect nucleotides with the correct ones, and DNA ligase seals the DNA back together. Now, any changes in the DNA that escape the proofreading and repair, because there's still errors, there are still DNA errors, which is errors in replication, which cause changes in the DNA code, and these are called mutations. And mutations are the actual source of any genetic variation. If we didn't have mutations in the DNA code resulting from errors in DNA replication, we wouldn't have any genetic variation and we wouldn't have any natural selection. We would have clones, and there would be no variety in traits, and nature couldn't select one trait over the other. So mutations are the source of genetic variation. They're the source of variety, and they're the source of the different traits that you find among different species, or among even individuals of the same species. The mutations are the source of genetic variety. And the reason we have natural selection, which is when nature selects the traits that are most suited for survival, is because of genetic mutations. Because we have mutations which give us a variety of traits for nature to choose from. There is a section on telomeres, and it's very interesting. And then the enzyme telomerase, which we've learned plays a role in aging. And I'm just going to encourage you to read that. So telomeres and telomerase. And then the only other thing I'll tell you is chromatin. Remember, okay, we learned that chromatin is the form of DNA when a cell is not actively dividing. So when the cell is in interphase, the DNA is loosely packed in a form called chromatin. And then when the cell is dividing, that chromatin condenses into chromosomes. So the loosely packed chromatin is called euchromatin. And that chromatin, when DNA is in this form called euchromatin, when it's loosely packed, then the DNA can replicate, and it can be used to produce proteins. The DNA code can be read, and then it can be transcribed into RNA and then translated into a protein, and protein synthesis can happen. But when the DNA begins to condense, before it becomes chromosomes, it condenses into heterochromatin. So it's just a more highly condensed form of chromatin. And that happens before it actually becomes a chromosome, okay? But when the chromatin is in the heterochromatin form, it's not able to code for anything. I mean, it's not able to be translated into a protein. So that's really, this chapter was just on DNA replication, and we're going to learn next in the next chapter about how DNA is transcribed into messenger RNA, and then that is translated into a protein. We're going to learn how DNA codes for proteins, which means that DNA codes for our traits. But this chapter was just on replication, and we're at the end.